Good day, I am Dr. Akinwunmi Adeoye. I will be talking about modulatory potential of citrus, sinuses, and Moringa, olivera hystrata, and Epiphytes on rat liver mitochondrial permeability transition, Pore. Mitochondria is one of the major organelles in the cell, and apart from its major metabolic functions in energy production, fire oxidative phosphorylation, it is also important in the initiation of apoptosis. Fluctuations of mitochondrial functions, such as permeability transition, has been found to play a crucial role in apoptotic process. Mitochondrial membrane permeability transition, Pore, is found in between the inner and the outer mitochondrial membrane and has been implicated in the development of several diseases. Some of the major components of this Pore includes the voltage-dependent anion channel, the adenosine nucleotide translocase, cyclophilin D, and other proteins that are found inside the Pore. Apoptosis is a programmed cell death, which is otherwise regarded as cell suicide. When a cell experiences insult or damage, the intrinsic mechanism in the cell will initiate repair mechanism. But when this repair mechanism could not reverse the damage or insult, then we have the process of apoptosis to be initiated. Apoptosis has two major pathways: the extrinsic pathways, which is otherwise known as death receptor pathways, where we have the ligand to bind with the cell surface receptor. And then the second pathway is intrinsic pathway, which has to do with mitochondria. One of the important factors that leads to the opening of mitochondrial membrane permeability transition Pore is oxidative stress. And we know that mitochondria is one of the sites where free radicals are being generated. Accumulation of these free radicals over time leads to oxidative stress. And if you have an increase in intracellular concentration of calcium coupled with the oxidative stress, then we have the Pore to be opened. We also have BCL2 family proteins that are important in the apoptotic processes, which will either be pro-apoptotic proteins and the anti-apoptotic proteins. When the concentration of the pro-apoptotic proteins are more than that of anti-apoptotic proteins, then it favors the release of cytochrome C from the inner mitochondrial membrane into the cytosol. The release of this cytochrome C is the point of no return for apoptosis to occur. The opening of this mitochondrial permeability transition Pore in the inner membrane leads to mitochondrial permeabilization and dissipation of membrane potential, which is followed by outer membrane rupture. The induction and inhibition of this Pore is important, and it has been implicated in several diseases. So too much of apoptosis will actually lead to neurodegenerative conditions. Why too little of apoptosis is also implicated, where we have uncontrolled cell growth, that is a cancer. So any agent that could induce the opening of this Pore will serve as an anti-cancer agent, and any agent that or compound that could inhibit or modulate the Pore opening could as well serve as agent that can relieve or that could relieve the symptoms of neurodegenerative conditions. Citrus sinuses is commonly referred to as sweet orange, and it is found to have a vitamin C, folacin, calcium, potassium, etc. While Moringa olivera leaves are used as vegetable in the preparation of soup and sauces in Nigeria and in many African countries. Epiphytes are organisms that grow on the surface of plants. They derive moisture, nutrients from air, from rain and water, and they are also important sources of food for many species. They differ from other parasites because they only grow on plants majorly for physical support, and they do not necessarily affect the host negatively. Mitochondrial permeability transition pore is an essential pharmacological target for the development of cytotoxic drug in the management of diseases with abnormal cell proliferation and where cell death is down-regulated. Justification of this study. Many dietary agents and many medicinal plants, they have become promising in the pharmacological targeting of mitochondria because of the secondary metabolite they contain and the bioactive compounds, which could either be modulator or inducer of the permeability transition. These natural compounds, they are isolated from fruits, from vegetables, and some of these medicinal plants have been proven to activate mitochondrial permeability transition pore via the mitochondrial mediated pathway of apoptosis. It is not known, it has not been reported whether the epiphyte of citrus sinensis and Moringa olivera will actually induce or inhibit the transition pore, the permeability transition pore. And so this study investigated the effects of citrus sinensis and Moringa olivera leaf extract and their epiphytes on MMPT pore in vitro. The specific objectives are to determine the in vitro antioxidant activities, to quantify some of the phenolic compound in the extracts using HPLC Dow array detector, and to assess mitochondrial swelling as well as assess cytochrome C release. Plant materials were collected, they were properly identified, and then extraction was done using standard procedures. Flavonoid content, total phenol, lipid scavenging ability, lipid peroxidation, further reducing antioxidant power, nuclear radical scavenging activities were determined. Compounds were quantified using HPLC. Mitochondrial swelling assay was done, as well as cytochrome C release. Data were analyzed by one way and another. Also, data were analyzed using graph-path prism, and the level of significance was set at P less than 0.05. The in vitro antioxidant potential of methanol leaf extract of citrus sinensis, Moringa olivera, and their epiphytes. All the extracts scavenged DBPH in a concentration-dependent manner, as well as nitric oxide radical. They also inhibited lipid peroxidation in a concentration-dependent manner. Table 1 is showing the high sensitivity value of extract in the in vitro antioxidant activities. While figure 4 is showing the total flavonoid, total phenolics, and FRAP potential of the extract. Moringa olivera had higher total flavonoid when compared to citrus sinensis and their epiphytes. While citrus sinensis extracts have had higher FRAP ability. Using the HPLC DAD to quantify the phenolic compound in the extracts, it was found that citrus sinensis it was found that tangeritin was more abundant in abundance in citrus sinensis, while eriocitrin is abundant in citrus epiphytes. But in Moringa olivera, moringinin was found in abundance, while flavone was abundant in Moringa epiphytes. Figure 5 is showing the induction of mitochondrial membrane permeability transition pore opening by calcium, and then the reversal by spermine, which is the standard inhibitor of mitochondrial membrane permeability transition pore. From this figure, calcium significantly induced the opening of this pore, while spermine inhibited the pore. The NTA is the non-triggering agent that is in the absence of calcium, while TA is the presence of calcium, and then spermine. The effect of varying concentration of these extracts on MMPT pore in the absence of calcium, which is actually to determine the effect of these extracts. It was observed that all the extracts inhibited the pore significantly in a concentration-dependent manner. But in the presence of calcium, when calcium was introduced, we have a mild induction, but will not be compared with that of calcium. In the same manner, Moringa olivera and its epiphytes also inhibited the pore in the absence of calcium. And as well, in the presence of calcium, we also have mild induction, which will not be compared with calcium. Cytochrome C release was found to be higher in the reference triggering agent, calcium. But in the presence of all the extracts, we have the release of cytochrome C is not as significant from that of NTA, that is in the absence of calcium. The findings from this research is that all the extracts inhibited DP peroxidation, scafenge, nitric oxide, and DPPH radical in a concentration-dependent manner. All extracts exhibited greater ferric reducing antioxidant potential. Results show that all the extracts possess substantive amount of total flavonoid and total phenolics. Citrus sinensis extract and Moringa olivera extract had higher total flavonoid and total phenolic content when compared with the epiphyte. HPLC data detector results revealed tangeritin as the most abundant in citrus sinensis extract, eriocitrin in citrus epiphytes, moringin in Moringa olivera extract, and flavone in Moringa epiphytes. All the extracts inhibited calcium in this opening of the pore in a concentration-dependent manner, with citrus sinensis leaf extract and Moringa epiphytes being the most potent in this regard. Based on the findings of this study, a significant relationship was obtained between antioxidant activity and phenolic content, indicating that phenolic compounds contribute significantly to antioxidant activity of citrus and Moringa. Thank you, and thanks to Academy of Excellence in Biosciences for supporting my registration.